Welcome to the deep dive. Ever wondered what's really going on inside your body? You know, beyond what the mirror shows.
It's a fascinating question, isn't it?
Absolutely. And today, we're not just wondering, we're plunging headfirst into a groundbreaking new way to actually see it all. We're talking about moving beyond those traditional kind of flat images
Right.
to a truly dynamic, information-rich 3D world of human tissue.
Okay, let's unpack this a bit. Imagine you're trying to understand a sprawling, bustling city just from a single flat map.
Mm-hmm.
You'd see the roads, the buildings, sure, but you'd completely miss the true scale of the skyscrapers, right?
Yeah.
Or the winding little alleyways, how different neighborhoods actually connect in 3 dimensions.
You wouldn't get the feel of it, the flow.
Exactly. You wouldn't grasp the intricate flow of life. And for centuries, when it comes to looking at our own tissues, we've largely been stuck with that single flat map. It's given us a lot, don't get me wrong, but it's left so much unseen. Precisely. For decades, the standard approach in disease diagnosis has been looking at these thin 2D slices of tissue under a microscope.
The H&E slides we often hear about.
Exactly, the hematoxylin and eosin stain slides. And while they've been incredibly valuable for spotting disease markers, this method, well, it inherently loses crucial spatial information just by its nature.
Right.
So the research we're diving into today, this is work from Johns Hopkins and NPOD. It introduces a really novel approach. It integrates advanced 3D imaging with something called nuclear segmentation.
Okay, nuclear segmentation, we'll get into that.
Yeah, and it allows us to literally reconstruct tissues in 3 dimensions. It gives us a far more comprehensive and, frankly, biologically accurate view than we've ever had before.
And our mission today, then, is to explore exactly how this advanced tech is uncovering patterns and interactions within our tissues that were previously invisible. Patterns that are absolutely crucial for understanding and ultimately, fighting diseases like cancer. This isn't just a technical tweak. It feels like a real paradigm shift in pathology.
It really is. It's potentially a shortcut to understanding these incredibly complex biological systems.
The hidden cost of 2D, what we've been missing.
So let's start with something fundamental. The cell's nucleus. We all learned in school it holds the DNA, but its physical characteristics, its shape and size, are actually incredibly revealing.
That's right. We talk about nuclear morphology, which is basically the size, the shape, even the texture of a cell's nucleus.
Like the cell's command center's appearance.
Exactly. And it's a key storyteller. Changes in these characteristics aren't just superficial tweaks. They often reflect really profound underlying changes. Genetic, epigenetic, even things happening in the microenvironment around the cell, all related to disease.
So it's like reading the expression on the cell's face.
That's a great analogy. A healthy nucleus tends to look a certain way, but a diseased one, it might be stretched out or swollen or just, dramatically misshapen. These subtle changes aren't just random noise, they're vital clues.
Okay, so if the nucleus is telling stories through its shape, what kind of critical stories are these changes telling us about, say, disease progression?
Well, they turn out to be powerful predictors of disease outcomes. For instance, studies have shown that heterogeneity, which just means the amount of variability,
The differences you see.
the differences in nuclear morphology within a single primary tumor can actually predict its metastatic burden.
Wow, so how likely it is to spread?
Exactly. Just based on looking at the variability in the nuclei, it's like a diagnostic fingerprint right there.
So for so long, those classic pink and purple 2D H&E slides, they've been the gold standard, right? The benchmark.
They have been, yes.
But what's the real rub here? What's the catch with relying only on those traditional methods? What are we missing?
The fundamental flaw, really, is that 2D images, just by being flat, they lose critical spatial information.
Okay.
Imagine trying to understand a really complex sculpture, but all you have are 100 incredibly thin slices of it laid out flat on a table.
Right, you see the detail in each slice.
You do, but you completely miss the overall form, the curves, the actual depth, how the different parts connect in 3D space.
You wouldn't know if a bump on one slice connects to something big or if it's just a tiny speck.
Precisely. And that's kind of what happens with 2D tissue samples. We get details, but we lose the context, the architecture.
It really is like trying to navigate a forest using just a single leaf.
Yeah.
And if we connect this to the bigger picture, like you said, disease progression, cancer cells invading, immune cells clustering.
Immune responses, changes in cell communication.
These are all fundamentally 3-dimensional processes, aren't they?
Absolutely. They happen in space.
Yeah.
So Relying only on 2D means we're just getting part of the story.
Yeah.
We could be completely misinterpreting crucial cellular interactions and how the tissue is really organized.
So, okay, quick recap for everyone listening. While the nucleus's shape tells us a lot about disease, our traditional 2D methods inherently miss that vital 3D spatial context. We're getting an incomplete puzzle picture,
That's a good way to put it.
which naturally leads to the question, if 2D is like peeking through a keyhole, what kind of universe opens up when we finally kick the door down and step into 3D?
Mm-hmm.
And that's exactly what these scientists have now built, which brings us perfectly to part 2, how they actually did it, how they made this new dimension a reality.
Building a new dimension, the 3D imaging pipeline.
So that question about stepping into the 3D universe, well, scientists at Johns Hopkins and NPOD have actually built the tech to do just that.
Yes, they have.
This paper introduces this novel and unified pipeline. It builds on an existing platform called CODA, right?
That's correct. CODA stands for Comprehensive 3D Digital Anatomy. It's essentially a platform for 3D cellular imaging, designed to quantitatively reconstruct large tissue samples, but at a resolution where you can still see individual cells.
No, it was already aimed at 3D.
It was, but this new research significantly extends its capabilities, specifically to overcome the limitations of those flat 2D images, and actually even some other 3D techniques. Things like light sheet microscopy, which can sometimes struggle with really large tissue volumes or introduce image artifacts.
Okay, got it. So how did they do it? Walk us through the process. It sounds incredibly complex.
It's definitely a multi-step process, but quite elegant. It all started with human pancreatic tissue.
From a donor.
Yes, obtained from a healthy organ donor through the NPOD network, that's the network for pancreatic organ donors with diabetes. Using real human tissue is obviously key for relevance.
Makes sense. So they have this block of tissue.
Yeah.
What next? Slicing.
Exactly. Imagine taking that block and slicing it incredibly thin. We're talking 4 micrometers thick sections.
4 micrometers. How thin is that?
It's roughly 1/10 the width of a human hair. Super thin.
Wow. Oh.
And they created hundreds of these ultra-thin sections. Then every 4th section was stained with H&E, that classic pink and purple stain, and then digitized at high resolution, 20x magnification.
So now you have hundreds of individual digital pictures, like pages in a book.
Precisely. And the first big computational step was aligning them all perfectly. It's called nonlinear image registration.
Okay, registration, what does that involve?
Think of it like this. You have these hundreds of digital slices, maybe slightly warped or shifted from the slicing process. Registration is about meticulously aligning each 2D slice with pixel-perfect precision, correcting for any distortions.
So you're digitally restacking them.
Exactly. You're digitally stacking them to perfectly recreate the original 3D tissue block, like digitally rebuilding that loaf of bread, slice by slice, but perfectly aligned. The result is a seamless, high-resolution digital 3D volume.
Okay, that makes sense. So you have the 3D structure rebuilt digitally. What's next? Identifying what's what?
Yes. That's where semantic segmentation comes in. First at the tissue level.
Semantic segmentation, meaning teaching the computer to recognize different tissue types.
Pretty much. They use the CODA semantic segmentation algorithm to label the major pancreatic tissue types. Things like the acini that make enzymes, the islets of Langerhans that make hormones, but also ducts, blood vessels, nerves, fat, and the connective tissue, the stroma.
And how accurate was that?
Impressively accurate. They achieved 94.9% accuracy in correctly identifying these different structures, all at a resolution of 1 micron per pixel. So essentially giving every tiny part of the 3D image a meaningful label. Context.
Okay, so that maps out the major structures, the neighborhoods in our city analogy, but what about the individual residents, the cells? How do they identify every single nucleus? That seems crucial.
Absolutely critical. For the nuclear segmentation at the cell level, they use a fine-tuned AI model called STARDIST. It's known for being good at finding nucleus-like shapes. They originally developed it for even higher magnification images, but they carefully adapted and optimized it for their 20x whole slide images.
And did it work well? Identifying millions of nuclei sounds tough.
It performed very robustly. They reported F1 scores, which is a measure that balances precision and recall of around 0.89, 0.84, 0.80 on different test sets, which basically means it did a very good job, high accuracy, in finding and outlining individual nuclei across the board.
So it found pretty much every nucleus correctly.
With high confidence, yes, guaranteeing we're seeing each cell in its proper place.
And then you mentioned earlier getting information from each nucleus.
Right. Once each nucleus was segmented, they automatically extracted a whole suite of features, over 20 different measurements for each nucleus.
Like what?
Things describing its shape area, circularity, aspect ratio, mean radius, but also its size, and even its stain intensity. They looked at the mean and standard deviation of the red, green, and blue color intensities from the H&E stain.
Wow, like a detailed biometric profile for every single cell's nucleus.
That's a good way to think about it, yeah. And the final, really crucial step was integration.
Bringing it all together.
Exactly. They registered the 2D coordinates of each identified nucleus directly into the 3D CODA volume they'd already reconstructed and segmented.
So you link every single nucleus profile to its exact 3D location and its surrounding tissue type.
Precisely. You know exactly where each nucleus is, what it looks like, and what kind of tissue it's sitting in, duct, eyelid, stroma, whatever.
So the outcome of all this meticulous multi-step pipeline, is these incredibly detailed 3D maps.
Yes. High-resolution 3D maps of nuclear morphologies, crucially preserving all those intricate spatial relationships. Between nuclei and between nuclei and the larger structures like ducts, nerves, blood vessels, it's a truly unified, comprehensive view.
Okay, let's do a quick recap for part 2. This team developed this amazing pipeline. Slice the tissue super thin, digitize it, digitally restack it perfectly in 3D, use AI to map out all the tissue types, use another AI to find every single nucleus, extract detailed features from each nucleus, and then link everything together in that 3D space.
That sums it up nicely.
So now we have this incredible detailed 3D map. The question becomes, what can we learn from it? What previously unseen patterns or behaviors could we possibly uncover about our bodies with this view?
The revelations, what 3D imaging uncovered?
And that question is exactly what the researchers tackled next. What they found was, well, pretty revealing. Let's talk about the first big finding. Just the sheer level of tissue and cellular heterogeneity they could now see.
Yeah, the scale itself was kind of staggering. The first sample they fully reconstructed and analyzed was roughly 16 by 14 by 2 millimeters. It doesn't sound huge, but...
How much tissue is that, actually?
Well, the volume was over 452 cubic millimeters. And within that volume, they identified and analyzed approximately 77 million nuclei.
77 million?
Million,
yeah.
Yes. Just to give you some perspective, that's easily over 100 times more cells than you'd typically analyze on a single standard 2D slide.
Wow. That's orders of magnitude more data.
Exactly. And it's that sheer scale that allowed them to move beyond assumptions you might make from looking at just a few small, potentially unrepresentative 2D snapshots.
So with that massive data set, what did they actually observe about cell density and nuclear shapes?
Well, they immediately saw significant heterogeneity differences in the tissue composition. For example, structures that are generally less metabolically active, like the stroma, the connective tissue, and nerves, they showed notably lower cell density, fewer cells per unit volume.
Okay, sparser areas.
Right. But in contrast, you look at the highly functional areas, like the acini, which make digestive enzymes, or the islets of Langerhans, making hormones like insulin.
Yeah.
Markedly denser, just packed with cells. It's like discovering these distinct ecosystems within the organ, each with its own unique cellular architecture and population density.
It's not just counting cells. It's like revealing entire cellular cities, each with its own vibe. And did the nuclei look different too?
Oh, absolutely. Huge variations in nuclear morphology depending on the tissue type. For instance, the nuclei in the islets of Langerhans. They tended to be very circular, like almost perfectly round on average.
Okay.
They showed considerable variation in their size, their area, some bigger, some smaller within the same islet.
Right.
And they had the largest average nuclear area overall.
Interesting. What about other cells?
Well, take acinar cells. They showed a more homogeneous size distribution, less variation in size compared to islet cells, still quite circular, but a bit smaller on average than islet nuclei.
And other tissues.
Cells in blood vessels, nerves, the ducts, the stroma.
Yeah.
They generally showed intermediate levels of heterogeneity in both size and shape. So each tissue type really had its own characteristic nuclear profile in 3D.
So this level of detail, this nuanced heterogeneity, it gives you a much more precise fingerprint for what healthy tissue looks like, right? Which could be huge for spotting early disease.
Precisely. It sets a much richer baseline.
Okay, finding number 2. This one sounds really important for understanding disease. Mapping immune cells, finding those immune hotspots.
Yes, this was a key application they demonstrated. Can we actually classify different cell types just using these nuclear morphology features we extracted?
Just from the shape and texture data.
Just from the nuclear features, yes. They trained a sophisticated AI model. It's called a random forest classifier. Think of it as using lots of decision trees together to make a really robust prediction.
Okay.
And they trained it to identify a specific type of immune cell, CD45 plus leukocytes, purely based on what their nuclei looked like in the H&E images.
And how well did that work?
Remarkably well. The classifier achieved about 89% accuracy in identifying these immune cells just from their nuclear appearance.
That's impressive. So they could map out the immune cells in 3D. What was so different compared to 2D?
This is where it gets really fascinating. The 3D analysis completely changed the picture of immune hotspots. You know, those areas where immune cells congregate.
How so?
Well, traditional 2D methods, because you're just taking sparse slices, they can really distort the spatial density and connectivity. Imagine seeing a few immune cells clustered on one slide. Are they really a dense cluster in 3D or just happening to line up in that one slice?
Right, it could be an illusion of the 2D view.
Exactly. But the 3D volume revealed much more localized and precise patterns. It showed exactly where these immune cells were truly clustered and presumably interacting.
And where were these true 3D hotspots located?
They identified leukocyte hotspots, primarily near stromal regions, blood vessels, and epithelial structures like the ducts. Conversely, cold spots areas with very few immune cells were mainly seen in the islets and nerves.
Okay, that makes sense. But was there anything really unexpected?
Yes. This was a critical discovery. When they did the quantitative analysis comparing immune cell density around different structures, they found a significantly higher density of leukocytes around larger pancreatic ducts compared to smaller ducts.
Larger ducts attracted more immune cells.
Significantly more. And similarly, though maybe less pronounced, there was a moderately higher density near larger blood vessels compared to smaller ones.
Whoa! So it's not just where the immune cells are, but their density depends on the size of the structure they're near.
Precisely. A size-dependent inflammatory response pattern. Immune cells preferentially gather around the bigger highways, the major ducts and vessels.
You would absolutely miss that in 2D, wouldn't you?
Completely. You might see some cells near a duct, but you wouldn't have the context to know if it was a big duct or small one, or systematically compare densities like this. It's an insight only possible with this comprehensive 3D view.
That's like finding rush hour traffic only jams up around major highways, not the small local roads. Game-changing insight for inflammation research or drug targeting.
It could be, yeah. It opens up new questions about why that happens.
Okay, and the third major finding you mentioned involved profiling the microenvironment around every single cell. What does that mean?
So because they knew the precise 3D location of every nucleus and the identity of the tissue surrounding it, they could analyze the neighborhood of each individual cell.
Its immediate surroundings.
Exactly. They did this by computationally generating spheres of different sizes, radii from 16 up to 128 micrometers, centered on each nucleus. And then they analyzed what was inside each sphere.
So you could see like what's right next to a cell versus what's in its slightly wider neighborhood and how that composition changes.
Precisely. And it revealed how the composition of that cellular neighborhood shifts depending on the scale you look at. For instance, if you looked at a small sphere around a nucleus in a dense acinar region, it would be mostly acinar cells.
Makes sense.
But if you took a nucleus near a rarer structure, like an islet or a nerve, the content within a small sphere might be dominated by that structure. But as you expanded the sphere outwards, the relative proportion of those sparser components would decline, while the more abundant tissues, like a sheeny, would start to dominate the volume.
So you get this dynamic view of the cellular neighborhood at different scales for every cell.
Yes. It allows for a much more sophisticated understanding of the context in which each cell exists and potentially interacts.
Okay, let's recap part 3 quickly. Finding 1. Massive scale analysis revealed unprecedented heterogeneity in cell density and nuclear shapes across different tissue types. Finding 2. 3D mapping uncovered the true nature of immune hotspots, revealing a previously unknown size-dependent inflammatory response around large ducts and vessels. Finding 3. They could profile the unique 3D microenvironment, the cellular neighborhood, around every single one of those 77 million cells.
That covers the key revelations, yes.
These insights are just profound. It makes you wonder, considering all this, what kind of medical advancements could realistically emerge from seeing our bodies in this completely new dimension?
The horizon impact, challenges, and the future of digital pathology.
As we look ahead, the broader implications, especially for medicine, seem huge.
They really are. I mean, this 3D approach gives us such a more comprehensive understanding of how tissues and nuclei are structured in space.
Seeing those patterns.
Seeing those spatial patterns, those interactions that were basically hidden before, offers insights that are just critical for understanding how diseases actually progress at a fundamental level.
So given these new capabilities, where do you see the most immediate impact? Which diseases or research areas stand to benefit the most right now?
Well, cancer pathogenesis is definitely a major one.
Cancer. Makes sense.
Yeah. In cancer, that interplay between the spatial organization of the tumor cells, how immune cells infiltrate the tumor, how the tumor interacts with the surrounding stroma,
Yeah.
it's incredibly complex and inherently three-dimensional.
Right.
Understanding these 3D relationships could totally unlock new ways to diagnose cancer earlier, predict metastasis much more accurately, and maybe even develop more targeted treatments.
It sounds incredibly promising, but technology like this, it never comes without challenges, does it? What are some of the current hurdles?
You're absolutely right. It's not trivial. One huge challenge is just the sheer computational demand.
Processing 77 million nuclei in 3D.
Exactly. Segmenting them, extracting features, analyzing the spatial relationships for millions, potentially billions of cells in larger samples. It requires serious computing power, robust hardware, very efficient algorithms. That's a significant bottleneck right now.
Makes sense. Data overload? Anything else?
Another point is the reliance on H&E staining. While it's the standard and tells us a lot about morphology, it does have limits. It doesn't give you deep molecular information about the cells.
Right, it tells you shape and general type, but not say what genes are active.
Precisely. It limits the granularity, the cellular features you can extract. It tells a big part of the story, but not the whole molecular story.
So where does it go from here? What are the next steps to build on this?
Well, that limitation actually points towards a really exciting future direction. It raises the question, what if we could combine this amazing spatial 3D data with molecular information?
Like layering other data on top of the 3D map.
Exactly. Integrating multi-omics data, things like genomics to see DNA mutations, transcriptomics to see gene activity, proteomics to see proteins with this 3D spatial context.
Yeah.
That could just dramatically enhance the precision and the biological relevance of the analysis.
So you'd know not just where a cell is and what it looks like, but exactly what it's doing molecularly, all within its 3D neighborhood.
That's the dream. Imagine that level of understanding.
Wow. What else is on the horizon?
Well, obviously, there's a need to validate this whole approach across many different tissue types, not just the pancreas, and across various disease models. Does it work as well in liver tissue or brain tissue or different types of tumors?
Testing its versatility.
Right. And then maybe the ultimate goal is to move beyond static snapshots and use this kind of framework to study dynamic processes over time.
Like watching things change.
Yeah, like watching how a tumor evolves, how it responds to treatment, or how an immune response unfolds in real time within the tissue's 3D structure. That would be incredible.
The journey, then, from that flat slide we started with to this full, explorable 3D landscape, it really isn't just a tech upgrade, is it? It feels like a fundamental shift in how we even approach diagnosis and treatment.
I think that's right. It's about seeing the unseen in a way that could truly matter for patients. Outro.
So to wrap things up, this 3D nuclear segmentation approach is clearly a powerful new framework. It's giving us unprecedented insights into the complexity of large tissues and the intricate mechanisms of disease.
It really sets the stage, I believe, for 3D imaging to potentially become the new benchmark, the new gold standard in digital pathology down the line.
So the next time you think about medical diagnosis, maybe picture this: that leap from a single flat static image to a vibrant, multidimensional, dynamic map of our own biology. How might that reshape the future of medicine for you, or perhaps for someone you know?
It really highlights something fundamental, doesn't it? Sometimes the most profound insights are found not just in what we see, but in how we choose to see it. The perspective changes everything.
A perfect thought to end on. And as always, we encourage you, our listeners, to stay curious about that vast, unseen world inside you, a world that science like this is bringing more and more into focus.